Here's part 5 of Chapter 14. So we left off in talking about epistasis and specifically when we're looking at coat color in a lot of mammals. So with Labrador retrievers, what we have here is one gene is going to actually express the color that would be present, either dark pigment or light, so either black or chocolate coloring. And then a second epistatic gene is actually going to determine whether that dark pigment is actually deposited in the fur. And if it is not, if you end up with a lack of deposit of this pigment, you end up with the yellow coloring in a lab. So what we say then is that the gene for pigment deposition is said to be epistatic to the gene that codes for black or brown pigment. Our next set of circumstances where Mendelian genetics really doesn't apply is the fact that we can have polygenic inheritance, which means that quantitative characters are those that vary in the population along a continuum. Polygenic inheritance means that we have more than one gene that works together to express the actual character. And when we have that, then we are setting up a situation where we can have a continuum of phenotype. So where we see that, for example, is with height. So there are a number of genes that work together to determine a person's height. And depending on what you have in terms of your alleles for these different genes will determine on how tall you become. Skin color is another example of a polygenic inheritance that is a quantitative character, which means we get variations along a continuum again. And this actually is an example of how that can work out and how that would look with this Punnett square. You can see it's a very complicated Punnett square. Oops, wrong. Where you can see your variation in skin color depending on how much of these different, how many of these different alleles you end up with. So in this case, we have the recessive alleles have little pigment, and the dominant alleles have a lot of pigment. And these two heterozygous parents, when they produce their gametes, this is what they end up with. So if you end up with a lack of pigment, you are very fair-skinned. But if you end up with a lot of pigment, if you get a lot of the dominant alleles between the crossing of these two gametes, then you end up with darker skin. So we have a continuum of everything in between there. Finally, what I want to look at is the effect of an environment on the phenotype. So this is another departure from Mendelian genetics, and that occurs when the phenotype for a character is also influenced by the environment. So we actually see that with hydrangeas, which is a really pretty flower that is influenced by the temperature in where the plant is located. And that temperature, that environmental factor, actually will determine what color the flowers end up being. So the same plant can actually express for anywhere from pink to purple to blue flowers, depending on what the environmental influence determines. So when we have a trait like that, we call that trait multifactorial. We already did some practice with pedigrees, and so I'm not going to spend much time on that, but I just want to review a little bit in case you have questions about what a pedigree is and what the basics of it include. So a pedigree is a family tree that describes the interrelationship of the parents and the children across generations. And inheritance patterns of specific traits can be traced and described using pedigrees. So we can use this for just looking at and following a trait through different generations. And we can also look at it, and it's used extensively when we're studying genetic mutations and disorders. So here, some of the basics that, just as a reminder, the male is indicated by a square, the female by a circle. When there is the phenotype expressing the trait, we say it's affected, then it's colored in. So if it's colored in, then that person has that trait. If it's not colored in, then the person does not have that trait. You also saw with the packet that you worked on that if there's a line drawn through the circles and squares, often that means that the person has died, is deceased. And if you have half of the square or circle, then sometimes that will indicate that that person is a carrier, so they're heterozygous for a recessive trait. Oftentimes, especially in more advanced biology classes, they actually don't show you the heterozygous, the carrier, being half shaded in. So they expect you to be able to deduce that based on the patterns that you see. I wanted to take a look at a few different examples of when genetics does not work right, or actually results in problems for the organism. So one of these is sickle cell anemia. So if you're not familiar with this disorder, it primarily affects African Americans or people of African descent. And it strikes 1 out of 400 African Americans. There's a very high frequency. You also find sickle cell anemia in Africa, so it's not just Americans who have this or experience this disorder. So this is an example of a disorder that's a result of pleiotrophy. So pleiotrophy is something we haven't talked about, but that happens when we have one gene that actually influences different and sometimes what seems unrelated phenotypic traits. Other examples of pleiotrophic genes affect phenylketonuria, which PKU is it's short for, which is a human disease that affects lots of different body systems but is caused by one gene defect. So if you have a mutation in a pleiotropic gene, you can have an effect on some or all of the traits at the same time. Other things, just again getting back to sickle cell anemia, what happens with sickle cell anemia is when we have the transcription and translation of the DNA sequences in making hemoglobin, there is one substitution mutation of a single amino acid, which causes the hemoglobin to then not actually fold correctly. So the protein, when it doesn't fold correctly, it actually makes this sickle shape here. And so this is a normal red blood cell, or that has hemoglobin in it. And here's one with the sickle mutation. And what happens is when oxygen levels are low, the sickle cell hemoglobin crystallizes into these long rods, and it deforms the red blood cells into this sickle shape. So we say that sickling, this effect here, creates a pleiotropic effect, which is, again, a cascade of other symptoms. The two alleles for sickle cell are codominant, which means they are both expressed. So you can have both normal and mutant hemoglobin at the same time being made in a heterozygote individual, so someone with both the dominant and recessive. So the sickle cell trait is recessive, but if you're heterozygous for it, you will appear to be somewhat normal. But you do have, because they're codominant, a person does have a little bit of the symptoms of sickle cell anemia. And usually what happens with someone with sickle cell anemia is whenever they have oxygen stress, um, the obviously their hemoglobin isn't working right, and so they're not able to get as much oxygen efficiently around their bodies, and so they get effects up from that, and it really results in a lot of pain. So because when the body is not getting the oxygen it needs, it sends out an alarm system saying, and that is the pain receptors. And often this is triggered when someone is in oxygen debt, which like during exercise. So someone who's heterozygous for sickle cell anemia, since it's codominant, we say that person has the sickle cell trait. They're not said to be a carrier, they're said to have the sickle cell trait because they actually do have sickle cell anemia. It's just not full blown because they have one normal hemoglobin allele. This is another picture showing how just this one amino acid change right here, is what results in these misshapen hemoglobin and red blood cells. And you can see what it comes down to on a molecular level is the difference between a hydrophilic amino acid and a hydrophobic amino acid, which causes the folding to be quite different in the two molecules. Okay, so another example of illness or disorder that is caused by things going wrong in genetics has to do with is cystic fibrosis. So in cystic fibrosis, I actually think we have our slides here in the wrong order. Yeah, we certainly do. Okay, so let me actually, before we go on to that, let me just move this out of where it's supposed to be. So let's go back and finish up with sickle cell anemia. So these are some of the symptoms of someone with sickle cell anemia, the sickle cell disease. And you can see how it would cause problems. And you can actually, if you're interested, pause this and take a closer look at each of these things. But one of the things I do want to point out is why is it that something like this continues on in a population generation after generation? And from a natural selection vantage point, is there has to be some sort of advantage to being heterozygous and carrying this trait, because to be homozygous recessive for this trait is a very debilitating disease. But to be heterozygous, actually what they have found protects a person from malaria. So in subtropical Africa, malaria is very common. And so that is why this is something that has become part of the genome of people of African descent, is because by having the heterozygous condition, having that one recessive allele for sickle cell disease, what they found is that people actually do not get the effects of malaria, do not experience them. So that is a huge advantage in a place where malaria is a much more detrimental and harmful disorder and disease. Okay, so now on to cystic fibrosis. Cystic fibrosis is the most common lethal genetic disease in the U.S. It actually strikes people of European descent, and 1 in 25 people of European descent is a carrier, which means they're heterozygous. So that tells you then cystic fibrosis allele is a defective or absent actually it's a defective allele which causes there to be an absence of chloride transport channels in the plasma membrane. These obviously, again, that allele is supposed to be making proteins that become chloride transport channels in a cell's membrane. And if they're not there, then there's a buildup of chloride ions outside of the cell. And that actually, there's a series of reactions that occur, but what ends up